One of the major focuses of our lab is studying pain pathways and understanding basically how drugs work that modulate pain pathways. And to do that, we either can use tissue culture or we can use cell lines to actually study how the drugs are actually interacting with the cells. As we bring the cells up out of liquid nitrogen, for example, we will put them into culture flask, and then we will literally give them artificial blood, so to speak, and media to grow. And so what we're doing in the tissue culture hood is we have to do it under sterile conditions so that we minimize contamination. But what we do then is we can actually culture them, which means that we can continue to grow them, so we can basically propagate them. But we also then use the tissue culture hood to actually transfer those cells to dishes so that we can actually do experiments with it. And the nice thing about these is that they're immortal in the sense that they continue to grow. And so we have a continuous supply of human cells. And the nice clinical relevance of that is that it is a human cell line. And then we're not having to, sacrifice animals. We actually are closest to the source of the disease. So once we have the culture dishes set up and the plates set up to actually do the experiment, a lot of times what we're going to be testing is how different drugs work. And what that means is we're going to be adding drugs to the cells and either waiting like 2 hours, 4 hours, or overnight. And then what we'll do is we'll actually look at through microscopy at what's going on inside the cell. So what we're doing is we're looking at either at proteins, specific proteins that are involved in disease progression, or we're looking at how a drug really regulates the expression of that particular protein. So what microscopy does is it allows us to actually get down at the cell level, and in some of the one microscope that we have in the lab, we actually can look inside the cells in living cells. So most of the time we're actually fixing the tissues of the cells so they're dead, and then we're just looking at a static picture. But we also have the capability to look at living cells and look at what's going on inside a living cell. One way to study protein expression is through the cell culture work. The more direct way and the more clinically relevant way is actually to look at saliva. So we look at proteins in tissues and these things, but actually looking at it in saliva is just another way to do it. What we do is we partner up with a lot of different organizations, clinics sometimes, and also universities. And what they do is we don't have the capabilities, because we're a basic science lab, to actually collect saliva samples. But the nice thing about the saliva samples is it's directly correlated with the disease progression, or it's tied into how they're responding to a different drug. And then what we can do is we're looking for proteins in the saliva to see if they're changing. It sometimes can be proteins, sometimes it can be things like prostaglandins or other inflammatory molecules. And then what we do is the refrigerator is actually filled up with samples from literally all over the country that we've obtained from the different clinical studies that we're working on. And then what we'll do with those is we have to actually work them up. So that means that we have to record the volume, we have to record how much protein is in it, log all that information and standardize it. One of the recent experiments that we were just working on, we generated almost 14,000 pieces of data just from the saliva samples that we obtained. So the overall goal that we're hoping to do is the translational component. How can we take the knowledge that we have and facilitate better patient care? So can we actually monitor disease progression in a better way? Can we figure out different drugs that are going to actually work to help treat that disease? And then can we monitor, really identify which patients are actually going to respond to those therapies?